Good afternoon. Thank you, the organization, for inviting me to present on behalf of Aspen today. I'm going to talk to you about our organization, the safety and tolerability and efficacy of our transplanted autologous derived dopamine neurons, precursors in moderate to advanced Parkinson's, but I will also discuss, that's our 6-month data in our lower cohort, but I'll also discuss a little bit about what we're doing in terms of manufacturing innovation. A little bit about our story. So we are headquartered in San Diego, and we are funded in 2018, starting with an early model that some preclinical data and ambitious dreams for every patient that would deserve their own cells. We are a company that was funded from the scientists from the Research Institute of Scripps Research in San Diego, and some patients from the Summit Stem Cell. Our vision is to use patient-owned cells to change the course of disease and change their lives for the better. So a little bit about Parkinson's. As you all know, there's a lot of companies developing treatments for Parkinson's. It is one of the second fastest growing disease in neurodegenerative space. There is about 90,000 people newly diagnosed a year in the US, and by 2050, there will be 12 million. It is an economic burden of about $7 billion for the US, and it is a loss of dopamine neurons. So most of patients that get diagnosed, by the time they go and get diagnosed, they have lost 50 to 60% of their dopamine neurons. And they are showing motor symptoms and non-motor symptoms. There continues to be an unmet medical need, and there's no drug that halts the disease progression. So what do we do? Our autologous product consists of dopamine neuron precursors that are implanted in the putamen, and they engraft, innervate, and create neuronal conditions that produce dopamine, aiming to treat motor symptoms of Parkinson's. You can see we implant those cells. It's a regenerative dopamine precursor, and we implant it in the putamen. They're A9-like dopamine precursors. A little bit about our platform. So our platform starts with small skin biopsy from the patient, and then we isolate the fibroblasts, and we reprogram those into iPSCs, and then we expand them into dopamine neurons, differentiate them, to dopamine neurons. One of the things that I'm really proud that we are very focused on is quality. We make sure that we have QC throughout the entire process. In process, we use also in silico QC methodology in order to make sure that we have the best cells to be delivered to patients. We have aligned with the FDA on requirements for our potency assay. Our approach is a matrix approach. It consists of 3 in silico and 1 orthogonal bioassay that establishes practicability in conversion of levodopa. We look to predict and identify the maturity of our neurons to ensure that we can predict their engraftment and their ability to release dopamine. We're also innovating in the manufacturing space to reduce the historical challenges with iPSC-derived manufacturing. We have an in-house 22,000 end-to-end manufacturing facility, and we cryopreserve we have 3 stages of manufacturing. We cryopreserve at every stage in order to enable us not only to be able to go back if we were to go back, but also to allow us to globalize and be able to distribute on a global level. As reference, we manufacture with a focus in quality, and we are well integrated at every stage of the product in order to reduce failure. We are now, with our partners, looking to and innovating in the manufacturing to achieve 90% process automation and using also machine learning. And we expect to have a scale-up in commercial capability to manufacture at least 250 patients per year as we scale up for our next pivotal study. Okay, a little bit about our program. As I mentioned to you, Okay, our SPIRIT trial is ongoing. We have actually announced last week that we're starting our third cohort, which is our commercial formulation. I will introduce to you today the 6-month data of our low-dose cohort. It has been completed, and we have shown that our cells, but also our occipital procedure has been successful. And although our trial is safety, and mainly safety and tolerability and dose response, we have secondary endpoints that I will show you today, showing preliminary glimpses of efficacy in these patients. So how do we deliver the cells? Here you can see a quick overview of the precision surgical approach. In a few slides, I will show you some of the early data. Using a thin cannula and the unique occipital approach, the therapy is carefully delivered under MRI guidance and deposit the cells at the posterior putamen exactly where intended. You can see they look like little pearls. Those are the deposits of the cells. There are 2 main lines, so we make a dorsal and a ventral deposit. And as you can see, this is the results of one of our patients in cohort 1. In terms of safety, to date, there's been no SAEs or unexpected AEs, and we have not observed any hemorrhages or graft-induced dyskinesias, and most patients are ready to be discharged within 24 hours. As demonstrated here, there are 3 out of 4 patients who received our therapy reported improvement on good on time, accompanied by reductions in off time. The average group improvement on good on time has been 1.7 hours, and the mean reduction of off time exceeds 2 hours. What I would like to call your attention is that out of the 4, 3 were well above the 2-hour mark for the good on time, and the same goes for the off time. As demonstrated, all 4 patients in a low-dose cohort have improvements all on PDRS Part 3 off scores. This is clear contrast with the gray line on top that we've matched with the natural history cohort from the PPMI database. We have seen at baseline a mean Part 3 score off was 42.3, and at 6 months, there was seen a reduction of 24.8. A group, I can actually read the slides here. Can you increase it? Sorry. Thank you. A group mean reduction of 17.5 points for this assessment of Part 3 off scores. I just wanted to also remind you that it is a 10-point score is considered a meaningful, highly meaningful difference. At 6 months, the change in patient-reported outcomes of activities of daily living and quality of life, and particularly, you can see that those activities, for the 4 patients, they showed a mean of 8.8 points, which relates to a 59% improvement. And at PDQ-39, which is an assessment of activity of quality of life, we have observed a mean of 5.7 decrease, a 44% improvement in these patients. In conclusion, at 6 months, our clinicians have reported and patients have reported that we have had an average improvement of 41% for the UPDRS Part 3 scores improvements in 59% of our Part 2 scores, and 44% in PDQ-39. We have achieved an average reduction of 2 hours in adjustment of time and a 1.7-hour average increase in adjusted good on time by Sousa diary. Precise intracranial delivery of our cells has been safe and well tolerated. And as I mentioned to you, patients are ready to be discharged 24 hours after the procedure. We continue to follow and evaluating a higher dose cohort. And as I mentioned to you, we are now moving to our commercial formulation, our cohort 3. I am specifically excited to be able to share the data in about 6 months' time, first half of next year, on our 12-month data for these patients and also the second cohort. I'd like to acknowledge the Aspen team, the patients, family members, and staff, our collaborators, and our investors that have joined us in our amazing journey to get these patients what they deserve, their cells, and improved quality of life. I have a couple of minutes. I think this is my last slide. I have a couple of minutes for questions. Well, thank you for your attention.
Thank you very much, Aspen Neuroscience.